Hello learners, welcome back to our channel Be Curious. Today we're gonna discuss very interesting topic human reproduction. Humans are sexually reproducing and viviparous. The reproductive event in human include formation of gametes, gametogenesis, sperms in males and ovum in females, transfer of sperms into the female genital tract, insemination and fusion male and female gametes leading to formation of zygote. This is formed by formation and development of blastocyst and its attachment to the urinary wall. Implementation. Embryonic development, gestation, and delivery of the baby, pasteurization. You have learned that these reproductive events occur after puberty. There are remarkable differences between the reproductive events in the male and in the female. For example, sperm formation continue even in old men, but formation of ovum ceases in women around the age of 50 years. The male reproductive system. The male reproductive system is located in the pelvic region. It includes a pair of testes along with accessory ducts, glands, and the external genital area. The testes are situated outside the abdominal cavity within a pouch called scrotum. The scrotum helps in maintaining the low temperature of testes 2 to 2.5 Celsius lower than the normal internal body temperature. Necessary for spermatogenesis. In adults, each testis is oval in shape with a length of about 4 to 5 centimeter and a width of about 2 to 3 centimeter. The testis is covered by a dense covering. Each testis has about 250 compartments called testicular lobules. Each lobule contains one to three highly coiled seminiferous tubules in which sperms are produced. Each seminiferous tubule is lined on its inside by two types of cell called male germ cells, spermatogonia, and spermatoly cells. The male germ cells undergo meiotic division finally lead to sperm formation, while Sertoli cells provide nutrition to the germ cells. The regions outside the seminiferous tubules called interstitial space contain small blood vessels and inertial cells or Leydig cells. Leydig cells synthesize and secrete testicular hormones called androgens. Other immunologically competent cells are also present. The male sex accessory ducts include rete testis, vasa efferentia, epididymis, and vas deferens. The seminiferous tubules of the testes open into the vasa efferentia through rete testis. The vasa efferentia leaves the testis and open into epididymis located along the posterior surface of each testis. The epididymis lead to the two vas deferens that ascends to the abdomen and loops over the urinary bladder. It receives a duct from seminal vesicles and open into urethra as the ejaculatory duct. These ducts store and transport the sperms from the testis to the outside through urethra. The urethra originates from the urinary bladder and extends through the penis to its external opening called urethral meatus. The penis is the male external genitalia. It is made up of special tissue that helps in extraction of the penis to facilitate insemination. The enlarged end of penis, called the glans penis, is covered by a loose fold of skin called foreskin. The male accessory glands include paired seminal vesicles, prostate, and paired bulbourethral glands. Secretion of these glands constitute the seminal plasma, which rich in fructose, calcium, and certain enzymes. The secretion of bulbourethral glands also helps in the lubrication of the penis. The female reproductive system. The female reproductive system consists of a pair of ovaries along with a pair of oviducts, uterus, cervix, vagina, and the external genitalia located in pelvic region. These parts of the system, along with a pair of the mammary glands, are integrated structurally and functionally to support the process of ovulation, fertilization, pregnancy, birth, and childcare. Ovaries are the primary female sex organs that produce the female sex gametes, ovum, and several steroid hormones, ovarian hormones. The ovaries are located one on each side of the lower abdomen. Each ovary is about 2 to 4 centimeters in length and is connected to the pelvic wall and uterus by ligaments. Each ovary is covered by a thin epithelium which encloses the ovarian stoma. The stoma is divided into two zones, a peripheral cortex and an inner medulla. The ovarian, fallopian tubes, uterus, and vagina constitute the female or accessory ducts. Each fallopian tube is about 10 to 12 centimeter long and extends from the periphery of each ovary to the uterus. The part closer to the ovary is the funnel-shaped infundibulum. The edges of the infundibulum possess finger-like projection called fimbriae, which help in collection of the ovum after ovulation. The infundibulum leads to a wider part of the oviduct called ampulla. The last part of the oviduct, isthmus, has a narrow lumen and it joins the uterus. The uterus is single and it is also called womb. The shape of the uterus is like an inverted pear. It is supported by ligaments attached to the pelvic wall. The uterus opens into vagina through a narrow cervix. The cavity of the cervix is called cervical canal, which, along with a vagina, form the birth canal. The wall of the uterus has three layers of tissue: the external thin membranous perimetrium, middle thin thick layer of smooth muscle myometrium, and inner glandular layer called endometrium that lines the uterine cavity. The endometrium undergoes cyclical changes during menstrual cycle, while the myometrium exhibits a strong contraction during delivery of the baby. The female external genitalia include mos, pubis, labia majora, labia minora, hymen, and clitoris. endometrium, myometrium, perimetrium, fibri, fallopian tubes. Most pubis is a cushion of fatty tissue or covered by skin and pubic hair. The labia majora are fleshy folds of tissue which extend down from the most pubics and surround the vagina opening. The labia minora are paired folds of tissue under the labia majora. The opening of the vagina is often covered partially by a membrane called hymen. The clitoris is a tiny finger-like structure which lies at the upper junction of the two labia minora above the urethral opening. The hymen is by a sudden fall or jolt insertion of a vagina tampon. Active participation in some sports like horseback riding, cycling. In some women, the hymen persists even after clitoris. In fact, the presence or absence of hymen is not a reliable indicator of virginity or sexual experience. A functional mammary gland is characteristic of all female mammals. The mammary glands are paired structures breasts that contain glandular tissue and variable amount of fat. The glandular tissue of each breast is divided into 15 to 20 mammary lobes containing clusters of cells called alveoli. The cells of alveoli secrete milk, which is stored in the cavities lumens of alveoli. The alveoli open into mammary tubules. The tubules of each lobe join to form a mammary duct. Several mammary ducts join to form a wider mammary ampulla, which is connected to lactiferous duct open at the nipple from which where the milk is expressed during breastfeeding. Gametogenesis: the primary sex organs that testes in the males and the ovaries in the female produce gametes, sperm and ovum, respectively by the process called gametogenesis. In testes, the mammal immature male germ cells (spermatogonia) produce sperms by spermatogenesis that begins at puberty. The spermatogonia, present on the inside wall of seminiferous tubules, multiply by mitosis, division, and increase in number. Each spermatogonium is diploid and contains 46 chromosomes. Some of the spermatogonia, called primary spermatocytes, periodically undergo meiosis. A primary spermatocyte completes the first mitotic division, leading to formation of two equal haploid cells, called secondary spermatocytes, which have only 23 chromosomes each. The secondary spermatocyte undergo the second meiotic spermatotide. The spermatocytes are transferred into spermatozoa sperms by the process called spermiogenesis. Spermiogenesis: after spermiogenesis, sperm heads become embedded in the spermatocyte cells. Embedded into Sertoli cell and are finally released from the seminiferous tubule by the process called spermiogenesis. Spermiogenesis. Spermiation. Spermiogenesis start at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormone. This is this, if you recall, is a hypothalamic hormone. The increased level of GnRH then acts at the anterior pituitary gland and stimulates secretion of two gonadotropin luteinizing hormone LH and follicle stimulating hormone FSH. LH act as the leading cells and stimulate synthesis and secretion of androgens. Androgens in turn stimulate the process of spermatogenesis. FSH act on the Sertoli cell and stimulates secretion of some factors which help in the process of spermatogenesis. Let us examine the structure of sperm. Sperm contain plasma membrane, acrosome on tip, nucleus containing chromosome material, head, neck, middle piece, mitochondria, tail. It is a microscopic structure composed of a head, neck, middle piece, and tail. A plasma membrane involves the whole body of sperm. The sperm head contains an elongated haploid nucleus, the anterior portion of which is covered by a cap-like structure acrosome. The acrosome is filled with the enzyme that helps fertilization of the ovum. The middle piece possesses numerous mitochondria which produce energy of the movement of tail that facilitates sperm motility essential for sperm fertilization. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for normal fertility at least 60% sperms must have normal shape and size, and at least 40% of them must show vigorous motility. Sperms released from the seminiferous tubules are transported by the accessory duct secretions of epididymis, vas deferens, seminal vesicles, and prostate are essential for maturation and motility of sperms. The seminal plasma along with the sperms constitute the semen. The function of male sex accessory ducts and glands are maintained by the testicular hormone androgens. The process of formation of a mature female gamete is called oogenesis, which is markedly different from spermatogenesis. Oogenesis is initiated during the embryonic development stage when a couple of million gamete mother cell oogonia are formed within each fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into phase one of the meiotic division and get a temporary arrest at the stage called primary oocytes. Each primary oocyte then gets surrounded by a layer of granulosa cells and is called the primary follicles. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. The primary follicles get surrounded by more layers of granulosa cells and a new therica and are called secondary follicles. The secondary follicle soon transfers into a tertiary follicle which is characterized by a fluid called filled cavity called antrum. The theca layer is organized into an inner theca interna and an outer theca interna. It is important to draw your attention that it is at least at this stage that the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division. It is an unequal division resulting in the formation of a large haploid secondary oocyte and a tiny first polar body. The secondary oocyte retains bulk of the nutrient-rich cytoplasm of primary oocyte. Thus, the first polar body formed out of the first meiotic division divide further or degenerate. At present, we are not very certain about this. The tertiary follicle further changes into the mature follicle or Graafian follicle. The secondary oocyte forms a new membrane called zona pellucida surrounding it. The Graafian follicle now ruptures to release the secondary oocyte from the ovary by the process called ovulation. This representation showing spermatogenesis A side in left side, oogenesis in right side. Spermatogenesis Spermatogonia goes through mitotic differentiation, give rise to primary spermatocytes. After it, after one's first meiotic division, it gives rise to secondary spermatocytes. After second meiotic division, it gives rise to spermatotite. After differentiation, it gives rise to spermatozoa. Oogenesis, oogonia, oogonia undergo mitotic differentiation, give rise to primary oocytes. After one first meiotic division completed prior to ovulation, secondary oocyte give rise to ovum. Menstrual cycle. The reproductive cycle in the female primate, monkey, apes, and human being is called menstrual cycle. The first menstruation begins at puberty and is called menarche. In human, female menstruation is repeated at an average interval of about 28 to 29 days, and the cycle of events starting from one menstruation till the next one is called the menstrual cycle. One ovum is released ovulation during the middle of each menstrual cycle. The major events of the menstrual cycle are shown figure. This diagrammatic representation of various events during menstrual cycle. Ovarian events, menstruation, follicular phase, luteal phase, next cycle begin. Developing follicles, maturation follicle, ovulation in 15 days, developing corpus luteum and regressing corpus luteum. The cycle starts with the menstrual phase when menstrual flow occurs and it lasts for 3 to 5 days. The menstrual flow results due to breakdown of endometrial lining of the uterus and its blood vessels which form liquid that comes out through vagina. Menstruation only occurs if the released ovum is not fertilized. Lack of menstruation may be indicative of pregnancy. However, if it may also be caused due to some other underlying causes like stress, poor health. The menstrual phase is followed by the follicular phase. During this phase, the primary follicles in the ovary grow to become a fully mature Graafian follicle, and stimulatively, the endometrium of an uterus regenerates through proliferation. These changes in the ovary and the uterus are induced by changes in the level of pituitary and ovary hormones. The secretion of gonadotropins (LH and FSH) increases gradually during the follicular phase and stimulates follicular development as well as secretion of androgen by the growing follicles. Both LH and FSH attain a peak level in the middle of cycle about 14th day. Rapid secretion of LH leading to its maximum level during the mid-cycle called LH surge induces rupture of Graafian follicle and thereby the release of ovulation. The ovulation is followed by the luteum phase during which the remaining parts of the Graafian follicle transform as the corpus luteum. The corpus luteum secretes large amount of progesterone, which is essential for maintenance of the endometrium. Such an endometrium is necessary for implementation of the fertilized ovum and other events of pregnancy. During pregnancy, all events of the menstrual cycle stop and there is no menstruation. In the absence of fertilization, the corpus luteum degenerates. This causes disintegration of the endometrium, leading to menstruation, making a new cycle. In human being, menstrual cycle ceases about 50 years of age, that is termed as menopause. Cyclic menstruation is an indicator of normal reproductive phase and extends between menarche and menopause. Fertilization and implementation during copulation, semen is released by the penis into the vagina. Insemination, the motile sperms swim rapidly past through the cervix, enter into the uterus, and finally reach the ampullary region of the fallopian tube. The ovum released by the ovary is called transported to the ampullary region where fertilization takes place. Fertilization can only occur if the ovum and sperm are transported simultaneously to the ampulla region. This is the reason why not all copulation lead to fertilization and pregnancy. The process of fusion of sperm with an ovum is called fertilization. During fertilization, a sperm comes in contact with the zona pellucida layer of the ovum and induces changes in the membrane that block the entry of additional sperms. Thus, it ensures that if only one sperm can fertilize an ovum, the secretions of the acrosome help the sperm enter into the cytoplasm of the ovum through the zona pellucida and the plasma membrane. This induces the completion of the meiotic division of the secondary oocyte. The secondary meiotic division is also unequal and result in the formation of a secondary second polar body and the haploid ovum otide. Soon the haploid nucleus of the sperms and that of the ovum fuse together to form a haploid zygote. One has to remember that the sex of the baby has been decided to this stage itself. You can know that the chromosome pattern in the human female is XX and that in the female XY before all the haploid gametes produce all the female have the sex chromosome X, whereas in the male gametes sperm the sex chromosome could be either X or Y. Hence, 50% of sperm carry the X chromosome while the other 50% carry the Y. After fusion of the male and female gametes, the zygote would carry either XX or XY depending on whether the sperm carry X or Y fertilize the ovum. The zygote carrying XX would develop into a female baby and XY could form a male. The mitotic division starts as the zygote moves through the isthmus of the aqueduct called cleavage toward the uterus and form 2, 4, 8, 16 daughter cells called blastomeres. The embryo with 8 to 16 blastomere is called a morula. The morula continues to divide and transforms into blastocyst as it moves further into the uterus. The blastomeres in the blastocyst are arranged into an outer layer called trophoblast and an inner group of cells attached to trophoblast called the inner cell mass. The trophoblast layer then get attached to the endometrium and the inner cell wall cell mass gets differentiated as the embryo. After attachment, the uterine cells divide rapidly and covers the blastocyst. As a result, the blastocyst become embedded in the endometrium of the uterus. This is called implantation and it leads to pregnancy. Pregnancy and embryonic development. After implementation, finger-like projection appear on the trophoblast called chorionic villi, which are surrounded by the uterine tissue and maternal blood. The chorionic villi and uterine tissue become interdigitation with each other and jointly form a structural and functional unit between developing embryo, fetus, and maternal body called placenta. The placenta facilitates the supply of oxygen and nutrients to the embryo and also removal of carbon dioxide and excretory waste materials produced by the embryo. Placenta also act as an endocrine tissue and produce several hormones called human chronic gonadotropin, HCG, human placental lactogen, androgen, progesterone. In the latter phase of pregnancy, a hormone called relaxin is also secreted by the ovary. Let us remember that HCG, HPL, and relaxin are all produced in women only during pregnancy. In addition, during pregnancy, all levels of other hormones like estrogen, progesterone, cortisol, prolactin, thyroxine are increased several-fold in the maternal blood. Increased production of these hormones is essential for supporting the fetal growth, metabolic changes in the mother, and maintenance of pregnancy. This figure shows that human fetus within the uterus. Cavity of uterus, placental villi, embryo, yolk sac, umbilical cord within the vessel. Immediately after implementation, the inner cell mass differentiates into an outer layer called ectoderm and an inner layer called endoderm. A mesoderm soon appears between the ectoderm and the endoderm. These three layers give rise to all this tissue in adult. It means to be mentioned here that the inner cell mass contains certain cells called stem cells which have the potency to give rise to all the tissue and organ. The human pregnancy lasts 9 months. Do you know how many months pregnancy lasts in dog, elephant, cat? Find out. In human being, after 1 month of pregnancy, the embryo heart is formed. The first sign of growing fetus may be noticed by listening to the heart sound carefully through the stethoscope. By the end of a second month of pregnancy, the fetus develops limbs and digits. By the end of 12 weeks first trimester, most of the major organ systems are formed. For example, the limbs and external genital organs are well developed. The first movements of the fetus and appearance of hair are the head are usually observed during the first month. By the end of about 24 weeks, the body is covered with fine hair, eyelid separate, and eyelashes are formed. By the end of 9 months of pregnancy, the fetus is fully developed and is ready for delivery. Pasteurization and lactation. The average duration of human pregnancy is about 9 months, which is called the gestation period. Vigorous contraction of the uterus at the end of pregnancy causes impulsion delivery of the fetus. This process of delivery of the fetus is called pasteurization. Pasteurization is induced by a complex neuroendocrine mechanism. The single signal for pasteurization originates from the fully developed fetus and the placenta, which induce mild urine contractions called fetal injection reflex. This triggers release of oxytocin from the maternal pituitary. Oxytocin acts on the uterine muscle and causes stronger uterine contractions, which in turn stimulates further secretion of oxytocin. The stimulatory reflex between the uterine contraction and oxytocin secretion continues, resulting in stronger and stronger contractions. This leads to expulsion of the baby out of the uterus through the birth canal. Pasteurization. The mammary glands of the female undergo differentiation during pregnancy and starts producing milk toward the end of pregnancy by the process called lactation. This helps the mother in feeding the newborn. The milk produced during the initial few days of lactation is called colostrum, which contains several antibodies absolutely essential to develop a resistance of the newborn babies. Breastfeeding during the initial period of infant growth is recommended by doctor for bringing up a healthy baby.